This chart plots the dementia rating scale against neocortical synapse density in Alzheimer's patients. The data yields a tight multivariate correlation of 0.96. Synapse loss is the strongest structural correlate of cognitive impairment in the disease. By comparison, plaque density contributes only 26% of that model's strength. The visible clumps of amyloid are a weaker predictor of dementia than the disappearing connections between neurons. To trace the actual execution of the disease, we have to look past the plaques and focus on the precise location where those connections fail, the dendritic spine of the excitatory pyramidal neuron. A dendritic spine is essentially a bag of actin. Its volume, and therefore the strength of the synapse it carries, is dictated entirely by a running mechanical balance between the severing of actin filaments and their stabilization. That physical balance is governed by a single protein, cofilin 1, which is switched on and off by phosphorylation at one highly specific node, serine 3. Because synapse loss drives dementia, any valid structural account of Alzheimer's disease must eventually explain exactly what happens mechanically at cofilin serine 3. This four-quadrant ledger tracks the current state of the literature. 4 independent laboratories have converged on this exact residue in Alzheimer's disease. They have not addressed each other in print, and their measurements create a massive physical contradiction. In 2013, researchers found that amyloid beta binds to an immune receptor called LILRB2. This interaction strips the phosphate from serine 3, causing destabilization and spine pruning. In 2018, a different group found that amyloid beta acts through a Rho-associated kinase, or ROCK. This pathway adds the phosphate to serine 3, which overstabilizes the actin network and impairs synaptic function. A third finding details the presence of pathogenic cofilin actin rods inside Alzheimer's neurites. The biochemistry dictates these transport-blocking rods mathematically require the absence of phosphate to form. Finally, a fourth independent lab corroborated the dephosphorylation pathway using a natural receptor antagonist against LILRB2 to block the effect. These are not conflicting murine models. Both the added phosphate finding and the removed phosphate finding were measured and verified in human Alzheimer's tissue. The field currently assumes one of these careful laboratories made an error, or that they are measuring different stages of disease. Standard linear biology cannot reconcile how a single node is driven in two physically opposite pathogenic directions at the same time. The paradox relies on a flawed biological premise. We assume cofilin operates as a linear on-off switch for actin stability. This graph plots actin stability against active cofilin, illustrating the curve established by Bamberg. Cofilin is a non-monotonic effector. The relationship turns over. On the left, dephosphorylated cofilin severs filaments. As occupancy climbs past the turnover point, it cross-links actin into pathogenic, transport-blocking rods. At the opposite extreme, if you drive phosphorylation up, cofilin is withdrawn from the filament entirely. The network overstabilizes, and the synapse loses the structural plasticity it needs in order to potentiate memory. There is no safe direction of travel from the healthy state. Both extremes destroy the synapse by different mechanisms. The laboratories are both correct about their isolated fractions. The disease state is not that cofilin is too active or too inactive. The effector has been driven off its functional center in both directions simultaneously. In a healthy dendritic spine, serine 3 is driven in both directions on demand. It acts as a transient local signal required to execute synaptic plasticity. The true physical lesion of Alzheimer's disease is a loss of this dynamic range. The residue is chronically pinned by unyielding ligand occupancy. Cofilin stops acting as a signal and becomes a fixed setting. Decades of pharmacological measurements have failed to notice this bimodal pinning because of how the data is collected. This diagram shows two populations of synapses. By relying on bulk tissue homogenate, researchers grind millions of distinct synapses into one sample. A cortex containing LILRB2-dominated synapses pinned low and ROCK-dominated synapses pinned high will average out to an uninterpretable, mathematically normal mean. The conflicting lab reports are artifacts of sampling. The reported direction depends entirely on which synapse population was slightly more abundant in their specific tissue fraction. This generates a strict, falsifiable prediction. Though the tissue's mean average may appear normal, the distribution of variants across individual synapses must be significantly wider. To prove this, researchers must analyze human excitatory synapses using array tomography to measure the per-synapse variance of phospho-serine 3. The predicted result is precise. Alzheimer's cortex will show a bimodal distribution of serine 3 variants, even in regions where the bulk mean is entirely unchanged. When a biological node is dysregulated, the pharmacological reflex is to block it. Rho-kinase, or ROCK inhibitors, are a current therapeutic class in active clinical development, intended to reverse the phosphorylation of cofilin. Returning to the U-curve reveals the mathematical hazard. Applying a monotonic, single-direction drug pushes every synapse leftward. The drug rescues the overstabilized synapses by pushing them to the healthy center, but shoves the already dephosphorylated synapses off the cliff into lethal rod formation. This mechanism dictates the clinical outcome. It creates the exact signature of a failed clinical trial with a positive subgroup, a pattern the field continually misreads as a dosing or staging error rather than a foundational biological conflict. The correct therapeutic target is not kinase inhibition or phosphatase inhibition. It is the restoration of signal-driven transient control over serine 3. The only characterized physiological system capable of delivering this transient control is the renin pathway, signaling through the ApoE2 receptor. This molecular schematic shows a strict biochemical staging requirement. Renin cannot act as a simple two-body ligand. It obligatorily requires a specific co-receptor to activate ApoE2, N-sulfated heparan sulfate. In a completely different sector of Alzheimer's drug development, efforts are underway to stop the transcellular propagation of tau protein using heparin mimetics. Pathological tau enters neurons utilizing the exact same heparan sulfate proteoglycans that renin requires to function. A heparin mimetic deployed to block tau will physically bind the polymer, inadvertently stripping renin from the cell surface by identical chemistry. This verified drug-level contradiction currently sits unacknowledged in the biopharma pipeline. Until we abandon bulk tissue homogenates, measures per synapse variance, and addresses this receptor conflict, we will continue to blind ourselves to the mechanical lesion of Alzheimer's disease.